Hello everyone, it's another episode on the microbial world where we learn the fundamentals of general microbiology. In this episode, we are going to be learning about fermentation. Kindly subscribe, share, like and drop your comments. Fermentation is a microbial metabolic process in which microorganisms convert organic substrates, most commonly sugars, into simpler final products such as acids, alcohols, and gases. This process occurs to extract energy, often in the absence of oxygen. Although it is commonly defined as anaerobic, it's important to note that some key industrial fermentations, such as acetic acid fermentation, are strictly aerobic. The process has been used by humans for millennia, for example, bread, beer, but it was Louis Pasteur in the 1850s who definitively proved that fermentation is a biological process carried out by living microorganisms. Modern biotechnology refines this by using pure cultures and highly optimized industrial bioreactors. Why microorganisms ferment? The nicotinamide adenine dinucleotide crisis. The central objective of fermentation, from a biochemical perspective, is not the production of alcohol or acid, but rather the regeneration of nicotinamide adenine dinucleotide. Cells primarily ferment to generate adenosine triphosphate energy in anaerobic environments. They must regenerate the electron carrier nicotinamide adenine dinucleotide, which is required for the initial energy-generating pathway, glycolysis. This process allows the cell to maintain redox balance and continue producing the small net yield of adenosine triphosphate from glycolysis, ensuring survival when oxygen is not available to receive electrons. Carefully study the glycolysis pathway. Types of fermentation. Fermentations are primarily classified based on their end products and oxygen requirement. 2.1 Classification based on end products. The type of fermentation depends on the specific enzymes that process pyruvate, the final product of glycolysis. Alcoholic fermentation, organisms, primarily yeast Saccharomyces cerevisiae. End products, ethanol and carbon dioxide. Applications, brewing, wine production, spirits, and bioethanol. Carefully study the alcohol fermentation pathway. Lactic acid fermentation lab, organisms, lactic acid bacteria like Lactobacillus and Streptococcus. Homofermentative pathway, produces lactic acid only, for example Streptococcus species. Heterofermentive pathway, produces lactic acid, ethanol, and carbon dioxide, for example. Leuconostoc species. Applications, yogurt, cheese, pickles, and sauerkraut. Acetic acid fermentation, organisms, Acetobacter aceti, Gluconobacter. Process, converts ethanol to acetic acid. Conditions, strictly aerobic, requires oxygen and is used for vinegar production. Carefully study the lactic acid fermentation pathway. Butyric acid fermentation, organisms, Clostridium species. Products, butyric acid, hydrogen, and carbon dioxide. Applications, historically used for solvent production, butanol, acetone closing parenthesis. Mixed acid fermentation, organisms, Enterobacterius, for example E. coli. Products, a complex mixture of acids, lactic, acetic, succinic, and gases, carbon dioxide, hydrogen. Classification based on oxygen requirement. Fermentations are often anaerobic, but the definition is broader in industry. Type of fermentation, oxygen requirement. Common organisms, aerobic fermentation, oxygen is required. Acetic acid bacteria, production of some amino acids. Anaerobic fermentation, no oxygen required or oxygen is strictly avoided. Yeasts, Clostridium species. Facultative fermentation can use either switches to fermentation when oxygen is depleted. Lactic acid bacteria, many Enterobacterius, biochemical basis of fermentation. Glycolysis, The common starting point: all major fermentation pathways begin with glycolysis, the process that splits one glucose molecule into two molecules of pyruvate, yielding a net gain of adenosine triphosphate and reduced nicotinamide adenine dinucleotide. The fate of pyruvate. The subsequent step differentiates the types of fermentation. In alcoholic fermentation, pyruvate is converted to acetaldehyde before becoming ethanol. In homolactic fermentation, pyruvate is directly converted to lactic acid. In both cases, this conversion serves the crucial purpose of oxidizing the reduced nicotinamide adenine dinucleotide back into nicotinamide adenine dinucleotide, thus restoring the redox balance and allowing glycolysis to continue. Industrial fermentation processes. Fermentation is scaled up using sophisticated technology to maximize product yield and quality. Components of a fermentation system. The fermentation takes place in a fermenter or bioreactor. This is a highly specialized vessel designed for process control, sterility systems, essential to prevent contamination by foreign microbes. Sensors continuously monitor pH, often controlled by base, acid addition, temperature controlled by jackets or coils, dissolved oxygen, dew, and foam levels. Medium requirements. The media provides the C, N, S, P sources, vitamins, and trace metals required for optimal growth and production. 4 point to phases and types of industrial culture. Industrial fermentation involves an inoculum development phase, scaling up the culture, a long production phase, and then harvesting, purification, and packaging. Industrial processes are often categorized by how the substrate and product are managed. Batch fermentation, a closed system where all components are added at the start. Simple and common for high-value products like antibiotics. Fed batch fermentation. Substrate is added during the process, for example to prevent toxic buildup or substrate exhaustion. Continuous fermentation, an open system where fresh medium is continuously added and spent medium containing product is continuously removed, allowing the culture to be maintained in the high-producing log phase indefinitely. Physical types of industrial fermentation. While the biochemical process remains the same, industrial fermentations are classified into two primary physical categories based on the growth medium. As submerged fermentation. This is the most common form of industrial fermentation. The microorganisms, for example yeast, bacteria, are grown in a liquid medium, completely submerged. Characteristics. The liquid medium contains all necessary soluble nutrients. Advantages. This method allows for easy control of temperature, pH, and oxygen transfer through agitation and sparging, and facilitates simple product recovery via filtration or centrifugation. Applications. Used for bulk products, pharmaceuticals, and all products requiring sterile, precise environmental control, such as antibiotics, recombinant proteins, insulin, and bioethanol. B. Solid-state fermentation. The microorganisms are grown on a moist solid substrate such as wheat bran, rice, or cellulose, in the absence of free water. Water is present only as moisture within the substrate. Characteristics. This method closely mimics the natural environment of many industrial fungi and bacteria, for example molds growing on decaying wood or food. The substrate often serves as both the nutrient source and the physical support. Advantages. SSF requires less energy, no heavy agitation, produces less wastewater, and often results in higher volumetric productivity for certain enzymes and complex aroma compounds. Applications. Used traditionally for koji starter for soy sauce, sake, certain enzymes, amylases, cellulases, and high-value aroma compounds. Products, factors, and applications. Fermentation drives multiple sectors of the bioeconomy, and its success is governed by controlling biological, physical, and nutritional factors. 5.1 Products of fermentation. Category Q. Products. Food and beverage: wine, beer, yogurt, cheese, bread, sauerkraut. Pharmaceuticals: antibiotics, penicillin, vitamins B12, vaccines, recombinant subunits. Industrial chemicals: ethanol, biofuel, butanol, acetic acid, vinegar, citric acid, enzymes, amylase, protease, lipase. Used in detergents, food processing. Factors affecting fermentation. Production yield is highly sensitive to the environment. Physical factors, temperature and pH directly affect enzyme activity and microbial growth rate. Agitation ensures uniform mass transfer of oxygen and nutrients. Nutritional factors. The carbon source type of sugar and nitrogen source form of amino acids must be optimized. Biological factors. Strain efficiency, the genetic quality of the organism and inhibition by end products, for example. Ethanol or lactic acid accumulation can poison the microbe itself.